Hey guys, hope you are doing well. You are welcome at Doctor's Digital College with Rashid Hashmi, having lectures of O levels and A levels for Cambridge Assessment International Examination, GCE, IGCSE, and AWS boards. Different subjects of metric, F.Sc, and anti-test will also be given here. They will also be included here. Hope you will learn here smart.
Welcome to Doctor's Digital College.
In this lesson, we will talk about strenous exercise or endurance and aerobics or moderate exercise. We will also discuss about what is aerobic and anaerobic respiration and what's the overall energy efficiency of anaerobic respiration. We will discuss that how is anaerobic respiration performed in different organisms. We will also discuss about what is the role of NAD, FAD and coenzyme A, and how can we use DCPIP and methylene blue or bromothymol blue as the redox indicator in laboratory conditions. Let's have an overview of aerobic respiration, overall process of aerobic respiration. You know that glucose is oxidized into pyruvate. This is done in the cytoplasm of the cell, and then pyruvate moves into the mitochondria, and whereby pyruvate is oxidized into acetyl coenzyme A. This is all we have already studied. And then acetyl coenzyme A enters into Krebs cycle. Okay, and this process is occurring in the mitochondria. While this process is occurring in cytoplasm of the cell. If it is here cell, I am representing the bigger box as cell and this smaller box as mitochondria. Then this is occurring like this. Okay, now let's see what are different products of these steps. You know that when glucose is oxidized, there is the formation of 2 net ATP, and during Krebs cycle, there is the formation of 2 net GTP. Okay, if we talk about in terms of NAD, during the oxidation of glucose, there is the net change of 2 NAD into 2 NADH2. During this process, once again, 2 NAD are oxidized, they are reduced into 2 NADH2. Okay, and during Krebs cycle, there is the formation of 6 NADH2, and there is the formation of 2 FADH2. Now, energy in terms of ATP which is readily usable or in the form of energy currency is this one, 2 ATP, or from these 2 GTP we can make 2 ATP. Energy stored in NADH2 or FADH2 cannot be readily used by the body. Now, until and unless it is oxidized by this process, that if it is NADH2 or if it is FADH2, it will be oxidized into H2O and plus ATP will be produced if molecular oxygen is available. Means NADH2 and FADH2, they will be oxidized. Their hydrogen will be oxidized into the water and ATP will be produced. But this process is possible only if oxygen is available. And if oxygen is not available, the whole of this process is just useless. The body is not doing oxidation of pyruvate or the oxidation of acetyl coenzyme A. Similarly, we know that fats and proteins, they are entering into this oxidation process at this stage. Fats and proteins, they are changed into acetyl coenzyme A, and then it will enter into the Krebs cycle. Okay, now once again, if there is no oxygen available or there is oxygen deficiency stress, fats are not oxidized. Beta oxidation of the fats is not proceeded. Now, if it is like this, then let's see what is the overall changes when we start exercise. And initially we are doing some heavy exercise, and after that when we will switch towards the aerobics, what will be the impacts on the body. Now let's see this graph. If on x-axis I'm drawing it time of the exercise, and on y-axis it is supply and demand of oxygen. Supply and demand of oxygen. During resting condition, if the demand of oxygen is this one, and the supply of oxygen and the demand of oxygen is this one, when we will start exercise, it will immediately rise to a higher level. Means the demand of oxygen means the demand of energy of the body will increase, so is the demand of oxygen of the body will increase to do aerobic respiration, and it will remain high until and unless we are doing almost the same level of exercise. And then when we will end exercise, it will come to normal once again, and it will be the same once again, like this. Now if we talk about the supply of oxygen, you know that supply of oxygen is not immediately increased up to the demand. Supply increases, supply of oxygen increases like this gradually. Means our breathing rate will increase gradually, our heartbeat rate will increase gradually, vasodilation of the blood vessels will occur gradually. So the supply of oxygen to the muscles will increase gradually, and a time may come when the demand and supply of oxygen, they are equal. Okay, and then when supply and demand of oxygen are equal, we are doing aerobics. This is our stress exercise. If I divide it into two parts, that this initial part of the exercise, I am writing here it as A. This A is the stress exercise, and after it, this B area is our aerobics, whereby oxygen supply and oxygen demand is equal. Body is not under the stress of oxygen deficiency. Okay, now when the exercise will be over, if I write here one arrow for the start of exercise and one arrow over here as the end of exercise, then when the exercise will be over, demand of oxygen will suddenly become normal. For the general metabolism, for the basic metabolism of the body, especially for the muscles activity, its demand will become exactly normal, which is during the resting conditions. But you know, our breathing rate will continue high, our heartbeat rate will continue to be high for some time, and the supply of oxygen will be gradually decreasing like this, and then it will become normal. Now if you look at the two areas, this area and this area, they are complementary to each other. They are compensating for each other. This area is called oxygen debit. This area is called, oxygen deficit, while this area is called oxygen debit. You might have listened to the words credit and debit in your banking matters. In this condition, body is under the stress of oxygen deficiency. It means body is coping with this oxygen deficiency somehow or the other. Okay, and this coping with this management is by the consumption of acids of the body. You know, in our body there is stored oxygen, there is stored ATP, there is stored GTP. All those acids are consumed to fulfill the demand of energy to the body, because supply is not up to the demand by aerobic respiration. So to fulfill this deficiency, the acids are consumed. Okay, I'm going to write here the acids are in the form of stored ATP, stored GTP, and stored oxygen. These three things are used, plus there is anaerobic respiration. That body will be doing anaerobic respiration in this stress condition. All these stored acids will be consumed. Okay, now when the exercise is over, now we have to repay these acids. We have to restore our stored form of ATP, we have to restore GTP, we have to restore the oxygen, stored oxygen within our myoglobin. Similarly, during aerobic respiration, some stress metabolites are produced like lactic acid, and that lactic acid is to be oxidized. For that purpose, there is some extra oxygen supplied even after the end of exercise, and that is called oxygen debit. This oxygen debit is basically to repay oxygen deficit. Now, overall time period for stress exercise can be different, and overall time period for this aerobics can be different. If we are doing heavy exercise like weightlifting or some competition in swimming or fast running in 100 meter, 200 meter sprint, until and unless we are doing stress exercise, demand and supply of oxygen will not be equal, and the body will remain under oxygen deficiency stress, and this part of respiration will not be done. So we can say there will not be the oxidation of the fats and the oxidation of NADH2, FADH2. Now make one point clear here, that in this stress condition there is not anaerobic respiration only occurring. There is aerobic respiration, there is anaerobic respiration. Aerobic respiration is occurring up to the extent oxygen supply is there, and for the deficiency of oxygen, for this little bit energy deficiency, there is anaerobic respiration. So this process is occurring to lesser extent, but along with it there is anaerobic respiration. Okay, but when we will enter into the aerobics, if we are not doing too much of heavy exercise, or we slow down our exercise after just warm-up exercise, then we will enter into the aerobics, and in aerobics, oxygen demand and oxygen supply, they are equal. Means body is doing overall this process and body is not doing this process. What does it mean that body is consuming fats and body is respiring carbohydrates completely? There is complete oxidation of the carbohydrates. Okay? And moreover, we can do this kind of exercise for a longer period of time, because in stress exercise, this lactic acid produced, this causes fatigue, or this causes pain in the muscles. Okay, and because of lactic acid, we have a limited tolerance to this lactic acid, and because of this limited tolerance, we cannot continue stress exercise for a longer period of time. On the other hand, aerobics can be continued for a longer period of time because body is not under stress. So I can say during aerobics, overall consumption of the carbohydrates is more. There is complete consumption of the carbohydrates, and there is also the consumption of fats and proteins. Now, what happens to this lactic acid? Lactic acid is basically changed into acetyl coenzyme A if oxygen is available. When oxygen will be available after exercise, it will be converted into acetyl coenzyme A. This is here acetyl coenzyme A. And you know that acetyl coenzyme A will come into Krebs cycle and it will be oxidized. Or there is also the fate of acetyl coenzyme A that it is changed back into glucose. Means from acetyl coenzyme A, the whole of this process is also reversed. Okay. Similarly, this acetyl coenzyme A formed from the lactic acid is converted back into fats. Means this process can be reversed. So we were talking about that during stress exercise, lactic acid produced is changed into acetyl coenzyme A, which will be then converted back into glucose or into fats, or it is also oxidized into carbon dioxide and water. Okay, and to do all these processes, there is the need of some extra oxygen, which we are doing even after exercise in the form of oxygen debit. Now, from whole of this situation, it is clear that when we are doing stress exercise, we are not consuming it. But when we are doing aerobics, we are consuming more of the fats. Through different experiments, it has been studied that when we are doing aerobics, there is a shift of this thing, that fats to lean tissues. It has been studied that when aerobics is done, more of the fats are consumed and less of the lean tissues are consumed. And overall, more lean tissues are developed. What is meant by the lean tissue? Lean tissue means the connective tissues of the body, connective tissues which are there in our muscles, in our blood vessels, the tissues which are attaching our muscles with our bones, and so on. They are giving overall elasticity to the body. So I can say that by aerobic respiration or by aerobics, body composition is shifted in the favor of lean tissues. So it is clear that when more elastic tissues will be there, overall efficiency, overall elasticity of the body will increase, blood vessels will be able to dilate more properly, they will be able to coil up more properly. Okay, overall breathing system becomes more efficient because in this condition we are rising our breathing system and cardiovascular system gradually to a higher level, and as the body composition is changed, their efficiency is increased. So through experiments it has been studied that by aerobics, overall efficiency of our breathing system, overall efficiency of our cardiovascular system is increased. Blood pressure is lowered because of more elasticity of the blood vessels, and as a result there is a lower chance of cardiovascular diseases or heart related diseases. Okay, and so these are the benefits of aerobics. On the other hand, as total amount of carbohydrates converted into lactic acid per unit time is much higher than in aerobics, and moreover fats are consumed, fats are produced as a result of stress exercise. So I can say more of strength is developed by stress exercise or during endurance. When we are doing heavy exercise, we are developing more power and more strength. Overall total amount of energy consumed by the body per unit time is much higher. Okay, although this is being done for a shorter period of time. Now, what are the impacts? Through a series of experiments, it has been studied that when overall consumption of energy per unit time is increased, it's boosted up, then confidence level of the individuals is increased. Okay, and there is peace of mind developed. As a result of confidence and peace of mind, there is better sleep developed after endurance. Okay, and there is more fortitude of the mind also. Mental stress is decreased. So we can say that this is the exercise of young age, when we need to develop confidence, when we need to develop strength, when we need to develop fortitude of the mind, when we need to have a better sleep. At that time this is needed. On the other hand, when we are in the mid of our age, like in late 40s or in early 50s or late 50s, we need overall maintenance of body composition. We need overall maintenance of elasticity of the body. Okay, we need to avoid stress, we need to avoid cardiovascular diseases, we need to avoid the rise in blood pressure, and for all such conditions there is aerobics more suitable. So in conclusion, I can say aerobics is the exercise of mid-age or late 40s, late 50s. On the other hand, endurance is the exercise of young age. Both kind of exercises, they are good. I cannot say that for overall maintenance of the body, for overall fitness of the body, one exercise or the other exercise is more suitable. Although they are more suitable for different age groups, but in general conclusion, I can say both type of exercises are needed for overall general fitness of the body. Now, if overall this process is clear to you, that what's happening in endurance and in aerobics. Now let's see how is anaerobic respiration done to produce this lactic acid. During this stress condition, how is glucose changed into lactic acid rather than to be changed into carbon dioxide and water? How is that done? Now let's see. So when oxygen deficiency is there, along with aerobic respiration, there is anaerobic respiration. And how is that done? You know that glucose is oxidized into pyruvate or pyruvic acid. Pyruvic acid is CH3, C double bonded O, C double bonded O, OH. Sometimes we write it pyruvate when this H is removed, or sometimes we write it pyruvic acid when this H is attached. And you know that during this process, there is the reduction of 2 NAD into 2 of NADH2. There is the formation of 2 ATP. We are getting 2 ATP. Now, if oxygen deficiency is there, we cannot convert NADH2 into NAD, meaning oxidation of nicotinamide adenine dinucleotide into oxidized form is not possible, because its oxidation is done by a molecular oxygen which we are inhaling into our body. Now, if it is not possible, how could we continue this process? The first step, which is called glycolysis, cannot be continued if we do not have oxidized form of NAD, because NAD here is acting as oxidizing agent for the oxidation of glucose. It will oxidize the glucose, it will help oxidation of the glucose, and itself it is reduced. We will later on discuss about the role of NAD overall, role of FAD and coenzyme A. Now, to continue this process, actually we convert pyruvic acid into lactic acid. It is like this, that pyruvate is converted into lactate or lactic acid. Now, look at its structure. Pyruvate is CH3, C double bonded O, C double bond OOH, while lactate is CH3, CO, C double bonded OOH. This one, and along with it there is one hydrogen attached over here and one hydrogen attached over here. So pyruvate is reduced into lactate by the addition of two hydrogen. Pyruvate is this is the reduction of pyruvate. If it is so, we have to reduce pyruvate into lactate. There must be some reducing agent. Now the reducing agent is NADH2, which itself will be oxidized back into its NAD. Okay, so this is a 3-carbon compound, this is 3-carbon compound, and pyruvate is reduced into lactate and NADH2 is oxidized into NAD. Now both of these processes are occurring within the cytoplasm. In case of aerobic respiration, NADH2 is actively transported into the mitochondria, whereby it will be oxidized by the molecular oxygen into NAD. But in this case, which is called anaerobic respiration, or it is also called lactate fermentation or lactic acid respiration, NADH2 is oxidized within the cytoplasm. Mean these two processes, they are occurring side by side within the cytoplasm. Now, let's see what is the energy efficiency of anaerobic respiration. You know that aerobic respiration, theoretically we got 38 ATP per mole of glucose. And here for anaerobic respiration, we have got just 2 ATP per mole of the glucose. Means it's obvious that overall energy efficiency of aerobic respiration is much higher than overall energy efficiency of anaerobic respiration. But let's have a different view. If we talk about energy efficiency as the number of ATP molecules or the number of moles of ATP produced per unit time. In case of aerobic respiration, it is here anaerobic respiration, it is greater than aerobic respiration. It is opposite to this, that total number of ATP molecules produced per unit time. I am writing here ATP per unit time, and this is here ATP per unit time. Per unit time ATP produced by anaerobic respiration is much higher. Why it is so? Because NAD is oxidized within the cytoplasm and these two processes they are occurring side by side. So overall speed of glycolysis is greater in anaerobic respiration than overall speed of glycolysis in aerobic respiration, because in aerobic respiration NADH2 will move into the mitochondria by active transport, and through electron transport chain it will be oxidized, and then oxidized form of NAD will be actively transported out of mitochondria and then it will be used in glycolysis. So overall energy efficiency of anaerobic respiration is greater per unit time than overall energy efficiency of aerobic respiration per unit time. And you know, during stress exercise, when we are doing heavy exercise, we are not concerned about the amount of glucose being consumed. We are concerned about the amount of energy produced per unit time, because that is an emergency situation, that is a stress situation. We need a quick supply of energy. That's why at that time more ATP per unit time is required, and for that purpose anaerobic respiration is more suitable. Now lactate or lactic acid is causing fatigue or pain in our muscles. Now I have one question to you, that if there are two individuals, for example me, I am not an expert athlete, I am just a common person who is not doing some heavy exercise or even not doing some walks, general daily walks, and there is another person or the student of A level who used to play soccer daily, and he is, you can say, or she is an expert athlete. He or she is used to do heavy exercise daily. Now, if we have the competition, me and that person who is doing exercise on daily basis, we have a sprint of 200 meter. We start. Then in the body of which of the two individuals anaerobic respiration will be started further? In my body or in the body of the student who is an expert athlete? This is a question to you. You can pause this video here and you can think about it, that what should be the answer. Okay. Now, let's see. Your answer might be that anaerobic respiration will be started earlier in the body of a non-athlete person or in my body, because I am not well adapted for, my muscles are not well supplied with oxygen. But actually the answer is other way around. Lactate fermentation or anaerobic respiration will be started first in the body of an expert athlete. I will start anaerobic respiration, say for example after 200 meter, and the expert athlete will start anaerobic respiration after 150 meter run. Why it is so? Actually, there is a scientific reason behind the early start of anaerobic respiration or lactate fermentation in expert athletes. Because this production of lactate in expert athletes help a higher level of aerobic respiration. Means there are not only the harmful effects of lactate fermentation on the body, means it's not only causing pain and fatigue to the body, it has some good and positive effects on the body. Let's see what are those positive effects. Lactic acid, although it's acidic thing and it is causing pain and fatigue in the muscles, but you know that being acidic, it will cause, number one, vasodilation. Vasodilation is done by the breakdown of H bonds, hydrogen bonds. They are broken down because of the acidity. So within the walls of blood vessels, hydrogen bonds are broken down between the connective tissues so they can dilate more. So more blood supply can be given to the muscles. Okay. Moreover, when more blood will be given to the muscles, exercising muscles, there is more uptake of heat from the exercising muscles. You know that during stress exercise, during heavy exercise, we need to remove heat because accumulation of the heat within the muscles can stop exercise, can stop doing exercise by the muscles earlier than the amount of pain produced by the lactate. So the removal of heat from the exercising muscles is more critical than the tolerance of lactate. Okay, so number two, it is better supply of oxygen. Lactate causes right shift of oxygen dissociation curve. This is oxygen dissociation curve in normal condition. If there is some acidic condition in the blood, there is oxygen dissociation curve is shifted towards right. This is ODC, means the release of oxygen from the hemoglobin becomes easier when the blood is more acidic. So lactate is causing acidity in the blood, so the release of oxygen from the hemoglobin becomes easier. This is what you have studied in detail in your previous classes. Now the third thing is there is lactate tolerance. That in the muscles of an expert athlete, overall lactate tolerance is increased, which increases overall the size of the muscles. When more lactate can be accumulated within the muscles and muscles can tolerate it, then overall size of the muscles is increased. So because of these good effects of lactate on the muscles of an expert athlete, anaerobic respiration or lactate fermentation is started earlier in the body of an athlete than in the body of some non-athlete person. Now let's see how is anaerobic respiration done in some microbes. Lactate fermentation or lactic acid fermentation is done in mammalian mussels or in some lactate bacteria. There are certain bacteria which are also producing lactic acid. They are called lactate bacteria, and they are also exploited in some industrial processes for the formation of lactate. Anaerobic respiration in microbes. Microbes like Saccharomyces, yeast group, and some bacteria, and roots of the plants, especially the tips of the plants where there is high demand of energy, they are sometimes doing anaerobic respiration when oxygen deficiency is there. Now in that case, the first step of glucose oxidation is same, that is called glycolysis. Glucose is oxidized into pyruvate, 2 ATP are produced and 2 of the NAD are reduced into 2 NADH2. Then this pyruvate is changed into this compound. This is called ethanol, or this is called acetaldehyde, and then acetaldehyde is changed into ethanol. Now the difference is that one H is attached over here and one H is attached over here. Means ethanol is reduced into ethanol. This is the reduction of ethanol, and here you can see that this is 2,3 carbon compound. This pyruvate is 1, 2, 3 carbon compound, while ethanol is 2 carbon compound. So there is the formation of 1 carbon dioxide along with it. Okay, this carbon dioxide is coming out from this one. This carbon dioxide is given out in this form, and ethanol. You know that when ethanol is to be reduced into ethanol, there is the need of some reducing agent, which itself will be oxidized. So the reducing agent here in this case is NADH2 is oxidized into NAD. Okay. This is reducing agent which itself is oxidized, and ethanol is reduced into ethanol. And you know, this ethanol is the waste product of microbes. It is excreted out of the body of microbes. While in case of plants, this ethanol is not the waste product. They are the higher or the advanced organisms. They will convert ethanol back into ethanol or acetaldehyde, and then acetaldehyde is changed into acetic acid, and there is a separate metabolic pathway for acetic acid oxidation process. So this is the overall process of anaerobic respiration in microbes or in plants, and this is also called alcoholic fermentation or ethanol fermentation or alcoholic anaerobic respiration. Okay, now let's see how some plants like Rhiza sativa, or you could say rice, is well adapted for anaerobic conditions. You know that rice need a high supply of water, means they are grown in waterlogged rice paddies. Even if the rice paddy is not waterlogged, we deliberately make it waterlogged, mean the water should be above the surface of the soil. This is the demand of that plant, that they need a high supply of water. Now, if it is waterlogged soil, oxygen trapped within the soil particles, in between the soil particles, they will be liberated. So, in the soil, there will not be oxygen available. But you know, roots have to do active transport for the active transport of, they have to do active transport for the uptake of mineral salts. Now, how can roots do active transport without respiration? So in that case, roots of the rice plant, they are doing alcoholic fermentation. They are doing this glycolysis, first step glycolysis, and then they are converting pyruvate into ethanol. Okay, now let's see how is the rice plant well adapted for overall anaerobic conditions of waterlogged soil. Rice or Rhiza sativa this is grown in waterlogged conditions where there is anaerobic conditions in the soil. So it has developed some basic adaptive features. Number one, it can do alcoholic fermentation. Okay, glycolysis is done, and after glycolysis, pyruvate is converted into ethanol. And number two, cells of the roots, they have high tolerance for ethanol. Means ethanol will be acting as inhibiting agent for certain enzymes, but the enzymes are well adapted in their cells that they are not inhibited by the ethanol produced. So the cells of roots of the rice, they have high tolerance for ethanol. And number three, they have high concentration of ethanol dehydrogenase. By ethanol dehydrogenase, ethanol is converted into acetaldehyde, COH is converted into CH3, CH2OH is converted into CH3, COH, and during this there is NAD is reduced back into NADH2. Okay, two of the hydrogen from the ethanol are removed, and then this acetaldehyde is changed into acetic acid, and then this acetic acid is oxidized in a separate pathway in some other area of the plant or when oxygen is available. Now this is metabolic adaptation of the root cells of the roots of Rhiza sativa. And the fourth one is they have developed parenchyma. Parenchyma tissue is developed in the stem of rice. What is parenchyma? You know that in the center of the stem there is pith. Pith cells are there. They are mostly for the storage of water and salts. Here in this case, in case of rice, these pith cells are damaged. They are broken down at certain areas and air spaces have been created. Now these air spaces are interconnected. They are continuous from leaves up to the roots. Just like the air spaces present in between spongy mesophilous cells, that some air space just inside the mitochondria is in connection with the other air spaces which are deep inside the leaf. So carbon dioxide entering through the mitochondria into some air space can go up to the depth of the leaf due to interconnection of air spaces. So is here that the air spaces of the stem of parenchyma in rice, they are continuous from the leaves up to the roots. So oxygen produced, NAD, FAD and coenzyme A. What is their role? You know that the full form of NAD is nicotinamide adenine dinucleotide, while the full form of FAD is flavin adenine dinucleotide, while CoA stands for coenzyme A. In the process of respiration, you have studied that whenever some substrate is oxidized, there is a need of some oxidizing agent. And whenever some substrate is reduced, there is a need of some reducing agent. Say for example, if this X is to be oxidized into Y, there must be some oxidizing agent. In this case, the oxidizing agent we have studied is this NAD, which itself is reduced into NADH2. There are different processes during which this oxidation process is occurring. I can write here one by one that there is, number one, during glycolysis, glyceraldehyde is oxidized into glycerate. Number two, during oxidation of pyruvate, pyruvate is oxidized into acetyl coenzyme A. You know, during Krebs cycle, citrate is oxidized into alpha ketoglutarate. Alpha ketoglutarate is oxidized into succinate. Succinate is oxidized into fumarate. Fumarate is oxidized into not fumarate, rather malate is oxidized into oxaloacetate. These are different oxidation processes which are occurring during respiration. And you know, in glyceraldehyde oxidation into glycerate, NAD is reduced. In the oxidation of pyruvate into acetyl coenzyme A, NAD is reduced. In the oxidation of citrate into alpha ketoglutarate, NAD is reduced. In the oxidation of alpha ketoglutarate into succinate, NAD is reduced. In the oxidation of succinate into fumarate, FAD is reduced. And in the oxidation of malate into oxaloacetate, NAD is reduced. Now, this is in glycolysis, this is in the link reaction within mitochondria, and all these four steps, they are occurring in Krebs cycle. Now, from all these steps, it is clear that there is dehydrogenation. Hydrogen is being removed. So the enzyme being used here is, in all these steps, there is dehydrogenase enzyme. It is removing hydrogen. Okay. If dehydrogenase is the enzyme, with it there is a coenzyme which is acting as prosthetic group. Complex organic compound which is not proteinic in nature. This is called prosthetic group. The prosthetic group of the dehydrogenase is NAD or FAD. These two are the prosthetic groups of enzyme dehydrogenase. So we studied that NAD is acting as a prosthetic group or the coenzyme for dehydrogenase enzyme. Whenever hydrogen is to be removed, they are taken away by NAD. So it's taken not only hydrogen, electron, but a lot of energy is there, which is given out during oxidative phosphorylation. Now let's see what is the structure of NAD and FAD. As I told that NAD stands for nicotinamide adenine dinucleotide. In it, the main role is of nicotinamide. This is the amide form of niacin. Niacin is vitamin B3. Vitamin B3 is the niacin and its amide form is nicotinamide. The structure of nicotinamide is like this. This is its amide group, NH2, and with it here it is one nitrogen attached. Now it's a resonance structure which has double bonds like this. 3 double bonds are there like benzene group, and with it is one hydrogen attached at this group. This is its R group. With it is attached with the dinucleotide. Okay, now I told that this is the key structure which is involved in oxidation and reduction process. Now this is its oxidized form. When it is reduced, what is its structure? The reduced form of it is that it will shift its double bonds like this. 3 double bond O, NH2. 2 hydrogens will be attached here and its double bonds will be shifted like this while the nitrogen is here. This is its reduced form and this is its oxidized form. This resonance structure can shift its bonds like this. Now with this part, the dinucleotide is attached. Let's draw how is that dinucleotide part attached. With this portion, this is ribose sugar attached, and here there are 2 phosphates attached with this ribose sugar. One is this phosphate, one is this phosphate. Okay, and with this 2 phosphates there is another ribose sugar attached. Okay, and with this ribose sugar, I have not drawn this ribose sugar of the same size. This is adenine nitrogenous base, and these 2 are the phosphates here. One phosphate is here, one phosphate is here. So this part is acting as one nucleotide and this part is acting as another nucleotide. So that's why this is called nicotinamide, and the same group is attached here. This is the key part acting as the active site of, just like active site of an enzyme. Overall, NAD is not an enzyme. This is a coenzyme acting as the prosthetic group of dehydrogenase. Okay. If it is so, then what is this FAD? I told that FAD stands for flavin adenine dinucleotide, and flavin is, in this case, the main part is of flavin, and this flavin is the derivative of riboflavin, and riboflavin is vitamin B2. Okay. And with its nucleotide is attached. Now let me draw what is the structure of dinucleotide here. At this point there is flavin part attached. This is here nitrogen, this is here nitrogen, this is here nitrogen, and this is here nitrogen. And there are 2 double bonded oxygens here. Okay, and it is double bond here, it is double bond here. So this portion is acting as resonance structure. Nitrogen is having 3 bonds. These double bonds of the nitrogen can be shifted to attach one hydrogen at each of them like this. If I draw it here once again, the double bond is shifted over here and the two hydrogens are attached like this. One hydrogen is attached at this point, one hydrogen is attached at this point, while this is its R group which is attached with this nucleotide. Okay, and this portion is as such, double bonded O, this is NH here as such. So this is its reduced form. The two hydrogens are attached with this resonance structure because of the shifting of double bond. And this portion is here attached as such. Now FAD, just like NAD, can uptake hydrogen like this in its reduced form, or it can release its hydrogen in its oxidized form. This is here plus 2H and this is here minus 2H. Okay, so in this way, in this form, it is acting as oxidizing agent, and in its form, it's acting as reducing agent when it is reduced as such. Now, let's see what is the next one, coenzyme A. Coenzyme A is the derivative of vitamin B5. It's a complex structure, but with it, there is sulfhydryl group attached with a complex structure. And now, coenzyme A is a carrier of acetyl group. What is acetyl group? You have studied that when oxidation of pyruvate is done, this acetyl is formed, CH3, double bonded O, this. Acetyl group is formed. This is a 2-carbon compound. Now, this 2-carbon compound is carried from oxidation of pyruvate to the Krebs cycle. This acetyl group can be attached with this sulfhydryl group of coenzyme A. Just like this, I am attaching it like this. This is here S, and now this is the rest part of coenzyme A, which is a complex. Now, so overall role of coenzyme A is the transfer of acetyl group from oxidation of pyruvate to Krebs cycle. We studied that NAD and FAD, they are acting as redox indicators. Now we can use artificial redox indicators to study that there is oxidation reduction process being done during respiration as well as during photosynthesis. For that purpose, we have the chemicals like DCPIP or methylene blue. If we talk about DCPIP, dichlorophenol, indophenol, this is a ring structure and it has resonance ring. Resonance ring is there so that it can attach hydrogen with it or it can remove hydrogen from it. And it's also called that it is a pH indicator. That in alkaline conditions, if there is slightly alkaline condition, it is blue. When it becomes neutral to acidic, it changes its color from blue to colorless to light pink. So when it will become, it is almost alkaline to neutral, it's in blue color. When it becomes slightly acidic, it becomes colorless, and then ultimately it becomes pH. You know that during oxidation process, there is the removal of hydrogen. The hydrogen which are carried away by NAD or FAD. If we are using DCPIP instead of NAD or FAD, it will be acidified and its blue color will become colorless or it will become slightly pink. So we can use DCPIP as a redox indicator, and the time taken for the disappearance of its color can be used as the rate of reaction. So if we are using some microbes like yeast or some germinating seeds or some small invertebrates, we can use a solution, slightly alkaline solution or solution of DCPIP or methylene blue, and then it will become colorless. And if we are able to measure the time, during which it becomes colorless, we can measure it as a rate of reaction, rate of respiration of that chemical or that microbe. Okay. Now, for this purpose, we can study the effect of temperature or the effect of different substrates, like how much quantity of some microbe like yeast is there, or how much quantity of the germinating seeds is there. Simply, it is done that in a test tube, we add DCPIP here, and this tube is closed. It is airtight or it is sealed. Inside it there is DCPIP plus there is some yeast or some germinating seeds are there. We add. And then we will start a stopwatch here. Okay, and we will measure the time taken for the disappearance of the blue color of DCPIP. Sorry, it is DCIP, not it is DCPIP. It is. Okay, so that will be the rate of reaction. We can change the temperature of this test tube by putting it in water bath. At different temperature, we can study the rate of respiration. Now, what are different factors which could affect this rate of respiration, and what is the overall protocol of this process of measuring the rate of respiration? We will study when we will be using simple respirometer for the study of rate of respiration. There I will tell you what different variables will be affecting this process and which are need to be controlled, and how will we perform it scientifically. We will need repeating it. We will need to remove anomalous values, and we will be doing it as a low risk experiment. And how will we measure the independent variable? How will we fix the dependent variable? Okay, how will we measure dependent variable and how will we fix independent variable? All these points of performing an experiment, designing an experiment and performing it will be discussed in the next topic, where we will use simple respirometer for the study of rate of respiration by germinating seeds or some small invertebrates. Here, my intention was just to tell you that DCPIP or methylene blue is acting just like NAD or FAD. They are acting as a redox indicator. That they, when they accept hydrogen, they become colorless. So is methylene blue. Similar to this, there is another one which is bromothymol blue. This is also a redox indicator. This is called BTB or bromothymol blue. It also acts as a redox indicator. Now, all these three, DCPIP, methylene blue and bromothymol blue, they can also be used as for the measurement of rate of photosynthesis, because in photosynthesis there is also oxidation reduction processes. Respiration is oxidation process and photosynthesis is overall reduction process. So we can use these for the measurement of rate of photosynthesis, which we will study in the next chapter. I hope that whole of this topic is very much clear to you, and you will like to have similar other topics of A levels and O levels with Doctors Digital College, and by subscribing it, so that you will be the first one to get its notification. Thank you for watching this video. Bye.